Now we shall talk about certain important concepts in the genetics which are important AIMS, JIPMAR, AFMC, EAMCET and COMEDK point of view. The most important points I'll put before you, dear students. Now the term genetics was coined by Bateson, the father of classical genetics. Is Gregor John Mendel. Father of classical genetics is Gregor John Mendel. Father of modern genetics is T.H. Morgan. Father of modern genetics is T.H. Morgan, Thomas Huxley Morgan. Okay, father of modern genetics. Morgan has conducted experiments mostly on Drosophila. Most of the experiments were conducted on Drosophila. So Drosophila is also called Cinderella of genetics. Drosophila is also called Cinderella of genetics. Now we shall talk about a concept called multiple alleles here now. Now what are alleles here now? A pair of genes present at corresponding loci of homologous chromosomes representing two different traits of a particular character can be called alleles. For instance, you take a pair of chromosomes here now, homologous chromosomes with a similar structure and similar position of centromere. There may be a gene at a particular site A, at the corresponding locus there is another gene called small a or maybe capital A. They are present at the same loci of two homologous chromosomes and both of them represent two different traits of particular character. What traits do they imply? For instance, you take capital A and small a, what traits do they imply? They represent the position of the flower. Capital A represents the axial flower, small a represents the terminal flower. Like if genes represent two different traits of particular character present at corresponding loci of homologous chromosomes, such genes are called alleles. Now in this, out of these two alleles, one is a dominant allele, the other one is a recessive allele. Recessive allele is formed by mutation of dominant allele. And in multiple allelism here now, more than two alleles are present at corresponding loci on different sets of homologous chromosomes, then it can be called multiple allelism. That is, for a particular character, if more than two alleles are present, and if these two alleles, more than two alleles now, if they are present at corresponding loci of different sets of homologous chromosomes, it can be called multiple allelism. Even in multiple allelism, a given organism carries only two alleles for a particular character. Okay, though they are formed by mutation of a normal gene here now, consisting of say about 5 to 6 alleles here now, but a given organism carries only a pair of alleles present at corresponding loci of homologous chromosomes. Like for instance, fur color in rabbit, is under the control of multiple alleles. Like there are alleles capital C representing the agouti trait, capital CH representing the chinchilla trait, CH representing the Himalayan trait, small c representing the albino trait. So all these are the different traits of the fur color of rabbit here now, agouti, chinchilla, Himalayan and albino. So how many alleles are there? 2, 4, 6, 8. 8 alleles are present here now. So these 8 of them can be referred to as multiple alleles. But for instance, if you take a given rabbit here now, always carries only a pair of multiple alleles here now, will never carry more than 2 alleles here now. Okay. Then, now by using the principle n into n plus 1 by 2, you can find out the total number of the pleiotrophic forms or total number of the morphological forms here now which exist in case of multiple allelism, where n is equal to number of alleles. N represents number of alleles. So if you substitute that here now, we will get the total number of possible morphological forms of particular character here now. Like for instance, if you take fur color in rabbit here now, how many alleles are there? 1, 2, 3, 4. So if you substitute 4 here now, 4 plus 4 plus 1 by 2, that is 20 by 2, that is 10. That is, it can exist, the total number of genotypic forms for this particular fur color can be about 10 here now. They can exist in 10 forms. Like the agouti exists here now in how many forms altogether here now? Okay, one agouti, then chinchilla, that is, it can exist in 4 forms, 1, 2, 3, 4. Same way, chinchilla can exist in 3 forms. Okay, Himalayan can exist in 2 forms. Albino exists in 1 form. So if you total up all these, 3 plus 2, 5, 9, 10. So altogether, how many forms it can exist? 10 forms, So by using the principle n into n plus 1 by 2, you can find out the total possible genotypic forms of multiple alleles, So in case of fur color of rabbit here now, there are 10 genotypic forms here now if you take that multiple allelism into account here now. Then same way, blood groups in human being is also an example for multiple allelism here now. Now there are A, B, O and AB blood groups in human being here now. The most important blood groups, there are some more rare type of blood groups also. The most commonly found blood groups among the people are A, B, O and AB here now. Among these, A, B and O were discovered by Carl Landsteiner, A, B, O were discovered by Carl Landsteiner, while AB was discovered by D. Cassillo and Sturley. AB was discovered by D. Cassillo and Sturley. So how many alleles are there here now? There are about, say, 3 alleles now. A allele, B allele and O allele. 3 allelic forms are there. Number of alleles here now. Number of alleles are 3 here now. So if you substitute in that particular principle n into n plus 1 by 2, you get, okay, around 6 allelomorphic forms. That is 6 different types of genotypic forms you get in the blood groups, Okay, if you substitute that particular 3. So you have 3 into 4, 12 by 2, that is 6. 6 different types of genotypic forms you find for the blood groups, Now the gene responsible for the blood group formation is present on chromosome number 9. Chromosome number 9. The gene responsible for imparting the blood group is present on chromosome 9. That particular gene synthesizes an antigen or immunogen. That particular antigen or immunogen responsible for the blood groups is present on 9th chromosome here now. So here this is the tabular form here now that tells us about the different types of antigens, antibodies present and the blood group. Always blood grouping is based on the type of antigen present. If a person carries antigen A, his blood group is A, and against this particular antigen a compatible antibody called B is present here now. The same way, a person having an antigen B, his blood group is B and he has got an antigen A. If both the antigens, A antigen and B antigen present, the blood group is AB and that person cannot afford to have any antibody with regard to the blood group here. And if the person has no antigens at all, but only, that is, antigens are nil here now, he has got both antibodies A and antibody B and his blood group is O group here. So in O group people, the antigens are absent, both the antibodies are present here now. Now this is the another tabular form here now, where it tells us about the mode of transfusion of blood groups. Suppose A group here now, A group can be given to A person, so there is no agglutination. There is no agglutination because antigen and antibody are compatible. A person has got antibody B, that's why antigen and antibody are compatible here now. A blood group cannot be given to B because B person has got antibody A, antigen A and antibody A are compatible, are incompatible, so agglutination takes place. Agglutination takes place. Now A can be given to AB because AB person does not have any antigens. A cannot be given to O because O person has got both the antibodies. So if A is given to O, there will be agglutination here now, that is plus plus. Here plus plus indicates agglutination, minus indicates no agglutination. Now B blood group, B cannot be given to A because A person has got antibody B, B antigen of donor, B antibody of recipient, both of them are incompatible. Agglutination takes place, so plus plus. Then B can be given to B, no agglutination. B can be given to AB, no agglutination. B cannot be given to O because once again it promotes agglutination here. Then same way you have AB. AB can be given to, it cannot be given to A because A person has got antibody B, antigen B of donor, antibody B of recipient, both of them are incompatible. Agglutination takes place. And AB cannot be given to B person because B person has got antibody A, antigen A of donor, antibody A of recipient are incompatible. Agglutination takes place. Then apart from this, AB can be given to AB because AB does not have any antibodies. AB cannot be given to O because O person has got both the antibodies. These two antigens will agglutinate both the antibodies, which proves to be very fatal. So plus plus. Then next you have O group here now. O group can be given to any person because O does not have antigen, so it does not induce any agglutination process. So minus, 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 minus. So on the basis of this, we can even fill these remaining columns also. A can be given to A, B. B can be given to B and AB here now. AB can be given to only AB here now. O can be given to all A, B, AB and O. That's why it acts as universal donor. Okay, O acts as universal donor. Suppose you get a question in the EAMCET here now, how does O act as universal donor? You should answer, O does not have any antigen, that's why it can be given here now. Now can receive from, A can receive from A and O. B can receive from B and O. AB can receive from, suppose you see this here now, vertical column, it can receive from A, AB, O and AB here now. A, B, O and AB. That is universal recipient. AB acts as universal recipient here now. Suppose you get a question in the EAMCET, how does AB act as universal recipient? You should answer, since AB does not have any antibodies here now, it can act as universal recipient. Now O can be given to, okay, O can be given to, O can receive from, O can be given to all, that is universal donor. You should write here. Okay, O can be given to all, A, B, AB and O. O can receive from only O here. If you see the vertical column here now, minus is present only in the O. Then plus plus indicates heavy agglutination. Apart from heavy agglutination, there is a provision for light agglutination also. Light agglutination is not fatal. Light agglutination is not fatal. Where are the chances for light agglutination in this tabular form? Suppose O, when it is given to A person has got antigen A. Antigen A of recipient can agglutinate antibody A of the donor. okay, which causes light agglutination. So here there is a chance for light agglutination which is not fatal. The same way O, when it is given to B person has got antigen B of recipient can agglutinate antibody B of the donor. So there is a chance for light agglutination even here. Now when O is given to AB has got both the antigens, A antigen and B antigen. Both the antigens of AB can agglutinate both the antibodies of O. So there is a chance for light agglutination even here. Now when what are the other chances? Say now when A is given to, say for instance AB has got both the antigens, A antigen, B antigen. Now A person has got antibody B. So B antigen of recipient can agglutinate B antibody of donor. So there is a provision for light agglutination even in this case. When B is given to AB, okay, B has got antibody A. So A antigen of recipient and A antibody of donor can agglutinate with each other, which may cause certain amount of fatality. Then so these are the chances where light agglutination takes place. Light agglutination is not fatal. Remember, it is not fatal because the amount of blood injected into the recipient from donor is very less, maybe hardly around 500 ml. In the amount of blood that is injected into the recipient, the amount of antibody present is very less here now. As a result, antibody cannot agglutinate all the RBCs of the recipient. That is why it is not fatal. Another reason for the light agglutination not being much fatal is when the blood is injected into a recipient here now, the antibodies present in the donor's blood get diluted. While being injected, they get diluted. As a result, they cannot cause heavy agglutination. So a light agglutination can be ignored and only heavy agglutination should be taken into account, and such blood groups should be avoided while transfusing the blood groups from donor to recipient here now. Now identification of blood groups here now. For this you have anti-A serum, anti-B serum. Now when anti-A serum is mixed with the blood with unknown blood group, here now, if there is a PPT with anti-A serum and with no PPT, then the blood is identified as I shall repeat once again. Look at this. Anti-A serum is mixed with the blood with unknown blood group. The blood is collected from a person whose blood group is not known. When it is mixed with this, if there is a precipitate, that is if there is agglutination causes precipitate formation. If there is a precipitate with the anti-A serum and if there is no precipitate with anti-B serum, the blood group is confirmed as A group here now, because the anti-A serum contains antibody A. You know very well that antibody A agglutinates antigen A. Suppose the blood group contains antigen A and antibody A, both of them interact with each other and thereby cause agglutination. That agglutination results in the formation of precipitate here, but there is no precipitate here. So if there is a precipitation only with the A antigen, then blood group is confirmed as A group here now. In the same way, anti-A serum is mixed with the blood of a person with unknown blood group. Now you have anti-A serum again mixed with the blood with unknown group. If there is a PPT, if there is no PPT with A and PPT with this, then it is confirmed as B group. Here in this, anti-A serum is mixed with the blood, there is no PPT. The same blood is mixed with the anti-B serum. The same blood is mixed with the anti-B serum. With anti-B serum there is a PPT. With anti-A serum there is no PPT. Then it is confirmed as B group here now. Because anti-B serum it contains antibody A agglutinates antigen B. Antibody B agglutinates antigen B. So in the second case here now, antibody B, that is serum contains antibody B, that antibody B agglutinates antigen B, so that there is a PPT formation, it indicates B group here now. The same way, anti-A serum is mixed with the blood with unknown group, there is a PPT with anti-A serum. With the anti-B serum also, there is a PPT. So if there is a PPT with both the blood groups, both the anti-serums, anti-A serum, there is a PPT, anti-B serum also there is a PPT, then blood group is confirmed as AB group. Blood group is confirmed as AB group because anti-A serum contains antibody A, anti-B serum contains antibody B, A antibody interacts with the A antigen, B antibody interacts with the B antigen, that is, it indicates that the blood that is collected from the person has got both A antigen and B antigen. That's why there is a PPT with both the groups here now. That's why it's confirmed as AB group here now. Then let's repeat this. Anti-A serum is mixed with the blood with unknown group, no PPT. Then anti-B serum is mixed with the blood with unknown group, here also no PPT, it indicates O group. So there is no PPT either with the A antiserum or B antiserum, it indicates that the blood group of the person is O group here now. How do you confirm that it is O group? Because the blood that is collected from here, it has neither A antigen nor B antigen. That's why it is mixed with either A antiserum or B antiserum. There is no PPT here now. Like that, the blood group can be confirmed in the lab here now by taking antiserum and mixing it with the blood of a person whose blood group is not known. Now another concept here now, RH antigen. RH antigen was discovered by Weiner and Levine. It was discovered by Weiner and Levine, and it indicates the presence of antigen in the human body. It was first detected in rhesus monkey, that's why it is named as rhesus factor. It was first detected in the rhesus monkey. Later on they found the similar antigen in human body also. That's why they named it as a rhesus factor in human body here now. In India, 95% of people are with rhesus factor. The people, those who do not have rhesus factor are called Rh negative. People, those who are with rhesus factor are called Rh positive. When there is an antigen D is present in the blood, it is referred to as Rh positive. And when antigen D is absent, the people, those who do not have this particular antigen D, it is referred to as Rh negative here. Rh negative. Now, unlike the normal blood groups here now, against antigen D, there is no natural antibody. Formation of antibody takes place in the recipient when their blood cells come in contact with the antigen D. Now, if Rh positive man marries a Rh negative woman, Rh positive man marries Rh negative woman, is a homozygous condition. The first child becomes Rh positive. When the woman becomes pregnant here now, the first child becomes Rh positive because positive is dominant over negative here now. Then again after 2 or 3 years, again when women conceives and carries a fetus here now, the second child is also Rh positive. Rh positive, but the second child dies of a condition called hemolytic disease of newborn. Dies of a condition called hemolytic disease of newborn. It is also called erythroblastosis fetalis. Erythroblastosis fetalis. It is also called erythroblastosis fetalis. Now what happens in this case, when the woman becomes pregnant first time, the child is Rh positive, that the child is carrying the antigens D. Now at the time of delivery, due to rupture of placenta, some of the RBCs carrying the antigen, Rh antigen, stealthily enter into the mother's blood and induce the formation of antibodies. So mother, soon after delivery, she develops RhD antibodies. RhD antibodies. She gets sensitized in response to that antigen and synthesizes RhD antibodies. So surplus number of antibodies are synthesized in her blood soon after first delivery. Again, when she becomes pregnant or she conceives after 1 or 2 years, the antibodies which have already been synthesized, all these antibodies enter into the second fetus through the placenta on a warfoot manner and damage the erythroblasts of the red bone marrow. Damage the erythroblasts of red bone marrow. As a result, second child dies of a condition called erythroblastosis fetalis. It is also called hemolytic disease of newborn. So this couple cannot have more than one child. Second delivery onwards, the children die. That is, whenever the woman becomes pregnant, after having the first child, all the children die of HDNB. However, it can be averted easily by injecting IgG immunoglobulins into her blood, okay, just before first delivery or soon after first delivery. If these immunoglobulin injections are given to the mother, these immunoglobulins destroy the fetal antigens, fetal RBCs. Whenever they enter mother's blood, they are destroyed so that mother does not get sensitized and the question of mother synthesizing the antibodies does not arise. So the formation of antibodies does not take place in her blood so that she can have any number of children. So it can be averted easily by diagnosing the blood group properly and by administering this particular immunoglobulin injection either just before delivery, first delivery, or the soon after first delivery, so that this particular immunoglobulin prevents the formation of antibodies in her blood. So this is the which one RH antigen, which was discovered by Weiner, Levin and Landsteiner, Weiner and Levin. And according to Race and Fisher, RH antigen, the genes responsible for RH antigen are present on chromosome pair 1. The normal blood group antigens are present on chromosome 9 whereas these antigens are present on chromosome pair 1. And they can exist in paired form like they can exist in capital C, small c, capital D, small d, capital E, small e. In this form, they exist at different loci on chromosomal pair 1. So depending upon the genotype, it can be confirmed. Suppose a person carries capital C, capital D, capital E, capital C, capital D, small e, small c, capital D, small e, capital C, capital D, capital E, he is confirmed as RH positive. Okay, if the person carries alleles like capital C, capital D, capital E and various combinations, is confirmed as RH positive. So among these, capital D antigen is responsible for imparting RH positive character to the person . So among these, D should be dominant. When the D is dominant, the person is RH positive, and when the D is recessive, the person is RH negative. I'll show you that. Capital C, small d, capital E, small c, small d, capital E, small c, small d, small e, small c, okay, capital C, small d, small e. So all these combinations, the person is referred to as RH negative. So in this condition, D is small, the rest of the antigens are capital letters, dominant. So irrespective of the dominant nature of the other two alleles, when the D allele is recessive, the person is Rh negative. Then where in this combination here now, the D allele should be dominant in order to contribute that particular character to the possessor. In this condition, see, you can make out here now, E is small here now, here C is small, E is also small here now, here, okay, all three of them are dominant, capital C, capital D, capital E. So in this condition here now, when allele D is dominant, the person is Rh positive, irrespective of the nature of the alleles of nature of other alleles, you know. In this case, when the D allele is recessive, the person is negative. So according to Race and Fisher, all these alleles are present on chromosomal pair 1. Then according to Weiner, there are 4 alleles here now which impart Rh characteristic feature to the human being here now. They are R1, R2, R0, RZ. So all of them impart Rh positive character to the human being here now. Then R0, R2 RY and RZ, they impart Rh negative character to the human being here now. So R1, R2, R0, RZ, they impart, okay, Rh positive character. That is, any of these 2 alleles when they are present here now, the person is Rh positive here now. Now R1, R2, RY, RZ, when any of these 2 alleles are present, the person is Rh negative. So according to Weiner, these are responsible for imparting Rh positive character, and according to Race and Fisher, these alleles are responsible for imparting Rh positive character to the human being here now. Now sex determination in human being here now and all other organisms. The X chromosome was called X body by Henking. Scientist Henking called X chromosome X body. And in human being here now, X human chromosomes, both autosomes and sex chromosomes, you know. Autosomes plus sex chromosomes. Sex chromosomes are also called allosomes, or they are also called heterosomes. So human chromosomes were discovered by Tizot and Levin. Human chromosomes were discovered by human autosomes, and sex chromosomes were discovered by Tizot and Levin here now. Now Y chromosome was discovered by Stevens and Wilson. Was discovered by Stevens and Wilson here now. Now sex is determined on the basis of the sex chromosomes here now, and there are several theories here which illustrate sex determination in human being and other organisms. Among these, the widely accepted hypothesis or concept, you can say, is chromosomal theory of sex determination. Chromosomal theory of sex determination. This chromosomal theory of sex determination is also called theory of heterogametes. This theory of heterogametes or chromosomal theory of sex determination was put forth by Carl Correns. Was put forth by Carl Correns. Now according to him, on the basis of the nature of chromosomes, the number of chromosomes here now, the sex of the organism can be determined. Now under this, first you have male heterogamy. Male heterogamy, that is male, instead of carrying 2 sex chromosomes here now, carries only 1 sex chromosome, which is called male heterogamy. Under this you have XX X0 condition, that is female is XX here now, male is X0, female is homogametic and male is heterogametic. female is homogametic, male is heterogametic. That is, when female produces ova, all the ova carry same kind of chromosomes, you know. That is, they carry same number of autosomes and same type of sex chromosomes. But when the male produces the sperms, you know, they carry haploid number of autosomes, but sex chromosomes, you know, some of them carry one sex chromosome and some of them do not have sex chromosome at all, because condition is X0 condition, you know. So if dissimilar gametes are produced with regard to the chromosomes, you know, you call it heterogamy. If identical gametes are produced with regard to the chromosomes, you call it homogametic. So female homogametic, male heterogametic, you know, this is found in cockroach, grasshopper, and locusts and bugs. This condition is found in cockroach, grasshopper, locusts and bugs, you know. Then second one is XX, XY condition. Female is XX, male is XY condition. This is noticed in human being and Drosophila. Is noticed in human being and Drosophila, XX and XY condition, where the women are with 44 autosomes plus X chromosome, 44 autosomes plus XX, and men are with 44 autosomes plus XY in case it is human being. And if it is Drosophila, 6 autosomes plus XX in female, 6 autosomes plus XY in male in case of Drosophila. So in Drosophila and human being, in both of them here now, females are homogametic and males are heterogametic. Human being, the total chromosomal number is 46 here now. In Drosophila it is 8 here now. So this is condition number 2. Female heterogamy. Female heterogamy is found in like Z0, ZZ condition. Z0, ZZ condition. It is found in Fumia, moths, butterflies. In Fumia is an insect here now. In moths and butterflies, you find Z0, that is females are heterogametic, males are homogametic here now. Females are heterogametic, males are homogametic. The same way, ZW females, ZZ males. This condition, again females are heterogametic, males are homogametic. This is found in insects, fish, birds and certain reptiles. Insects, fish, birds and certain reptiles, you know, where the female birds are ZW and the males are ZZ, you know. That is, cocks are ZZ, hen are ZW, you know. So in case of birds, you know, fish, insects and reptiles, you know, the fertilization is determined on the basis of the chromosome of the ovum itself. Okay, in case of human being, fertilization, the sex is determined at the time of fertilization, you know. Sex is determined at the time of fertilization. In case of human being, suppose the sperm with Y fuses with the ovum, that zygote is male. The sperm with X fuses with the ovum, that zygote is which one? Female here now. Whereas in case of the birds here now, and fish, reptiles and other animal reptiles and fish and insects here now, depending upon the nature of the chromosome of the ovum, the sex is determined. Suppose the ovum carries the chromosome W, it develops into male. If the ovum carries chromosome W, it develops into female here now. If the ovum carries the chromosome Z, it develops into male. If the ovum carries chromosome W, it develops into female. So you don't have to wait till fertilization, you know. Sex is determined on the basis of the chromosome of the ovum itself. And this kind of determination is called progamic type of sex determination. Progamic type of sex determination. And in case of human being, it is called syngamic type of sex determination. Syngamic type of, because you have to wait till fertilization. Only at the time of fertilization, sex of the fetus is determined here now. So like that, the female heterogamy is found in two conditions, condition number 1, Z0 and ZZ, Fumia, moths, butterflies are the examples. Second one is ZW, female, ZZ, male, found in insects, fish, amphibians and reptiles. Now in honeybee, the fertile females are called queens and the males are called drones. The fertile females are called queens and the males are called drones. Now the drones have haploid number of chromosomes, whereas queens are diploid with 32 chromosomes. Queens are diploid with 32 chromosomes. Now the queens lay two kinds of eggs. Queens lay two kinds of eggs. Two kinds of eggs. The eggs, some of the eggs get fertilized with the sperm. Some of the eggs get fertilized with the sperm to form a zygote with 32 chromosomes. Now if this zygote is fed with royal jelly, it transforms into queen. If the same zygote is fed with bee bread, it transforms into worker, which is a sterile female, and queen is a fertile female. Okay, it produces eggs. Some eggs fuse with the sperm, they give rise to the zygote with 32 chromosomes. If the zygote is fed with royal jelly, it develops into queen, which is a fertile female. If the same zygote is fed with bee bread, it develops into worker, which is a sterile female. The royal jelly contains salivary secretion of queen. It contains more amount of nectar and less amount of pollen grain. Whereas the bee bread does not contain salivary secretion of queen. However, it contains more amount of pollen and less amount of nectar. So because of more nutrition well-fed nutrition it develops into queen. Because of undernutrition, if this particular zygote develops into sterile female, that is worker. And now this ovum does not fuse with the sperm, it directly develops into drone through parthenogenesis. This particular ovum does not fuse with the sperm, it directly develops into drone through parthenogenesis. And there is a kind of haploid parthenogenesis which is called arhinitoki. This haploid parthenogenesis is known as arhinitoki. Now on the basis of this, it is fancifully stated that in honeybees, there are no fathers but they have grandfathers. Okay, honeybees, the workers, that is drones, drones have no father but they have grandfather. Drones have no father but they have grandfather. It is fancifully stated because this particular ovum does not fuse with the sperm, so it is stated that no father, no involvement of father, but however it has a grandfather because this ovum is produced by the queen who has got both mother and father. So with regard to sex determination in case of honeybee. Now in human being, apart from the chromosomes, sex of the fetus before birth itself can be determined by a method called amniocentesis. Amniocentesis technique, the amniotic fluid is collected after first trimester from the pregnant women and it is cultured and it is identified for the presence of bar bodies. Identified by the presence of bar bodies. What are bar bodies? Bar bodies are the heterochromatin substances heterochromatin substances present on X chromosome. In human being, the female carries two X chromosomes, whereas in male only single X chromosome is present. As a result of that, the genetic dosage of the female is high, genetic dosage of the male is low. because the presence of two X chromosomes, genetic dose of female is high, genetic dose of male is very low. So in order to bring about that equality between male and female here now, one of the X chromosomes in females is inactivated by a heterochromatin. The process of inactivation by heterochromatin is called dosage compensation. Dosage compensation. So dosage compensation is brought about by a heterochromatin that lies on one of the X chromosomes. For every two X chromosomes, so that there will be a bar body here now. Now this bar body in human being, in humans, it was discovered by Murray Barr. It was discovered by Murray Barr, and it was noticed in cats now, interface neurons of cats by Barr and Bertram. So in cats it was detected by Barr and Bertram, and in human being it was first noticed by Murray Barr and Kate Moore. In human being it was noticed by Murray Barr and Kate Moore, and in cats it was noticed in the interface neurons, in their interface neurons by Barr and Bertram. So it is a heterochromatin present on one of the X chromosomes, so that a kind of dosage compensation is brought about in both male and female here now. Now the process of inactivation of X chromosome by means of bar body is called X chromosomal inactivation. This X chromosomal inactivation concept was put forth by Mary Lyon and Lyanna Russell. It was the X chromosomal inactivation, which is also called lionization. Lionization technique was put forth by Mary Lyon and Lyanna Russell here now. Now the genic balance theory is the second theory. The first one is chromosomal theory of sex determination, as I made a mention earlier. It was put forth by Carl Correns. The second one is genic balance theory. This was put forth by Calvin Bridges. Calvin Bridges, and this theory is applicable to Drosophila. Is applicable only to Drosophila. Now according to him, in Drosophila, the autosomes carry certain genes which are responsible for determining the male characteristics. Autosomes carry certain genes which are responsible for determining male characters. X chromosome carry genes which are responsible for determining the female characters. So X chromosomes are responsible for determining female characters. Autosomes are responsible for determining male characters. Y chromosome has no significance in determining the sex. Y chromosome has no significance in determining the sex. Sex of male. However, Y is required for its fertility. There can be a male without Y chromosome also, but that is a sterile male here now. So in order to be fertile, the male requires Y chromosome. But Y is not essential for determining the maleness of the Drosophila. Now on the basis of this, Calvin Bridges has put forth a concept called sex ratio. Sex ratio is obtained by dividing number of sets of autosomes by number of sex chromosome, X chromosomes. Sex ratio is determined by dividing number of X chromosomes by number of sets of autosomes. number of X chromosomes is divided by number of sets of autosomes. So that gives you sex ratio. On the basis of sex ratio, the sex of the Drosophila can be determined. Now if X by A ratio is equal to 1, it indicates that the given insect is a fertile female. If X by A ratio is equal to 1, it indicates that the given insect is fertile female. If X by A ratio is greater than 1, if X by A ratio is greater than 1, that is about 1.5, it indicates that the given insect is super female, or also called meta female. If X by ratio is equal more than 1, it is super female or meta female, which is sterile here now. Then if X by A ratio is 0.5, it indicates that it is a fertile male. If X by A ratio is equal to 0.5, it indicates that the given insect is fertile male. If X by A ratio is 0.5 to 1, if X by A ratio is equal to 0.5 to 1, it indicates that the given insect is intersex. Given insect is intersex. So it shall repeat. If X by A ratio is equal to 0.5 to 1, it indicates that the given insect is intersex, which is also sterile here now. If X by A ratio is less than 0.5, it indicates that the given insect is super male or meta male, which is sterile. So according to Calvin-Bridges here now, by calculating the X by A ratio, and on the basis of X by A ratio, the sex of the Drosophila can be determined. X by A ratio is nothing but the sex ratio. It is deduced by dividing number of X chromosomes by number of sets of autosomes. If X by A ratio is equal to 1, it indicates the given insect is fertile female. If X by A ratio is equal to greater than 1, it indicates that the given insect is a super female or meta female. Then if X by A ratio is equal to 0.5, it is a fertile male. If X by A ratio is equal to 0.5 to 1, it is an intersex, which is sterile. And if X by A ratio is equal to less than 0.5, it indicates that it is a meta male or super male, which is sterile. And this concept is applicable only to Drosophila. So according to Calvin-Bridges here now, maleness of Drosophila is determined by genes present on autosomes rather than Y chromosome here now. So there can be a male even without Y, but in order to be fertile, that in the male, in order to be fertile here now, it has to have a Y chromosome.